Immunity is the body's defense mechanism against harmful substances. It protects us from bacteria, viruses, fungi, and parasites. The immune system recognizes and destroys invaders. It is a complex network of cells, tissues, and organs. Constantly on alert, it patrols the body for danger, launching a coordinated attack when threats are detected. The immune system is a remarkable testament to the human body's complexity. Macrophages and neutrophils are two types of white blood cells that play a crucial role in the body's innate immune response. They are among the first cells to arrive at the site of infection or injury, where they work tirelessly to engulf and destroy pathogens. Macrophages are large, phagocytic cells that are found in tissues throughout the body. They act as scavengers, engulfing and digesting cellular debris, microbes, and other foreign substances. Neutrophils are the most abundant type of white blood cell and are highly mobile. They are also phagocytic and have a short lifespan, often dying within a few days. Neutrophils contain granules filled with enzymes and other antimicrobial substances that help destroy pathogens. Upon encountering an infection, neutrophils release these substances, trapping and killing microbes. Both macrophages and neutrophils are essential for controlling infections and preventing the spread of pathogens. They are key players in the body's first line of defense, working tirelessly to keep us healthy. Their ability to quickly identify and eliminate threats is crucial for our survival. Without these dedicated cells, our bodies would be vulnerable to a constant onslaught of infections. Antigens are molecules that can trigger an immune response. They are like red flags that alert the immune system to the presence of something foreign or potentially harmful. Antigens can be proteins, carbohydrates, lipids, or nucleic acids. They are found on the surface of bacteria, viruses, fungi, and other pathogens. Each antigen has a unique shape that allows the immune system to recognize it. The immune system learns to distinguish between self-antigens, which are found on the body's own cells, and non-self-antigens, which are found on foreign invaders. This distinction is crucial for preventing the immune system from attacking the body's own tissues. Self-antigens are present on the surface of our cells and signal to the immune system that these cells belong to the body and should not be attacked. Non-self-antigens, on the other hand, indicate the presence of a foreign invader. When the immune system encounters a non-self-antigen, it mounts a targeted attack against it. This attack involves a complex series of events that ultimately leads to the destruction of the pathogen or infected cell. The primary immune response is the body's first encounter with a specific antigen. When the immune system encounters a new antigen, it takes some time to recognize it and mount a full-blown response. This initial response, known as the primary immune response, is typically slow and less effective than subsequent responses. During the primary immune response, B cells and T cells, two types of white blood cells, work together to eliminate the threat. B cells produce antibodies, which are proteins that specifically bind to the invading antigen. These antibodies can neutralize the pathogen, preventing it from infecting cells, or mark it for destruction by other immune cells. T cells, on the other hand, can directly kill infected cells or help activate other immune cells. A crucial aspect of the primary immune response is the generation of memory cells. Memory cells are long-lived B and T cells that remember the specific antigen encountered during the primary response. These cells provide long-term immunity, allowing the immune system to respond more quickly and effectively upon subsequent encounters with the same antigen. Section 5. Antibody Structure and Function Precision Engineering for Defense. Antibodies, also known as immunoglobulins, are Y-shaped proteins produced by B cells. They are the workhorses of the humoral immune response, targeting and neutralizing pathogens in a highly specific manner. The structure of an antibody is intricately linked to its function. Each antibody molecule consists of 4 polypeptide chains, 2 identical heavy chains, and 2 identical light chains. These chains are held together by disulfide bonds, forming a flexible Y-shaped structure. The 2 arms of the Y contain the antigen binding sites, which are highly variable regions that determine the antibody's specificity. Each antibody has a unique antigen binding site that can bind to a specific epitope on an antigen, much like a key fitting into a lock. This specific binding triggers a cascade of events that leads to the destruction of the pathogen. The tail region of the antibody, known as the Fc region, interacts with other components of the immune system, such as complement proteins and phagocytic cells. This interaction can trigger a range of effector functions, including complement activation, opsonization, and antibody-dependent cell-mediated cytotoxicity. Section 6. The hybridoma method producing monoclonal antibodies. Monoclonal antibodies are laboratory-produced antibodies that recognize a single epitope on an antigen. They are produced by hybridomas, which are hybrid cells created by fusing a B cell with a myeloma cell. Myeloma cells are cancerous plasma cells that can divide indefinitely in culture. The hybridoma method, developed in 1975, revolutionized the field of immunology and led to the development of monoclonal antibodies as valuable tools for research, diagnosis, and therapy. The process of producing monoclonal antibodies begins with immunizing an animal, typically a mouse, with the antigen of interest. This immunization stimulates the mouse's immune system to produce antibodies against the antigen. B cells from the mouse's spleen are then isolated and fused with myeloma cells. The fused cells, called hybridomas, are then screened for their ability to produce the desired antibody. Once a hybridoma producing the desired antibody is identified, it can be cultured indefinitely, producing large quantities of identical antibodies. Monoclonal antibodies have several advantages over polyclonal antibodies, which are mixtures of antibodies that recognize different epitopes on the same antigen. Section 7. Monoclonal antibodies in medicine, diagnosis, and treatment. Monoclonal antibodies revolutionize diagnosis and treatment. Their specificity targets cells or molecules, aiding modern medicine, used in assays to detect hormones, tumor markers, or infectious agents. Widely used in pregnancy tests to detect HCG in urine. Treats cancer, autoimmune, and infectious diseases by targeting specific proteins. Minimizes damage to healthy cells, reducing side effects. Section 8. Types of immunity, innate and adaptive. The immune system has two branches, innate and adaptive immunity. Innate immunity is the body's first line of defense, present from birth. It includes physical barriers like skin and mucous membranes. Adaptive immunity develops over time, recognizing specific pathogens. It is mediated by B cells and T cells, providing targeted attacks. Both systems work together to protect the body from infection. Section 9. Vaccines, stimulating the immune system for long-term protection. Vaccines are one of the most successful public health interventions in history. They have saved millions of lives and continue to protect people of all ages from serious diseases. Vaccines work by mimicking natural infection, triggering the immune system to produce antibodies and memory cells without causing the actual disease. Vaccines contain weakened or inactive forms of pathogens, or even just specific components of pathogens, such as proteins or sugars. When a person receives a vaccine, their immune system recognizes the vaccine antigens as foreign and mounts an immune response. This response is similar to the response that would occur during a natural infection, but because the vaccine antigens are weakened or inactive, the person does not develop the disease. The immune system then produces antibodies and memory cells that can provide long-term protection against the specific pathogen. If the person is ever exposed to the actual pathogen in the future, their immune system will be able to recognize it and mount a rapid and effective response, preventing or minimizing the severity of the disease. Section 10. Vaccination programs controlling infectious diseases. Vaccination programs have been highly effective in controlling and even eradicating infectious diseases. By achieving high vaccination rates within a population, we can establish herd immunity. Herd immunity occurs when a significant portion of the population is immune to a disease, making it difficult for the disease to spread. This protection extends even to those who are not vaccinated, such as infants, pregnant women, or individuals with weakened immune systems. Vaccination programs have led to the eradication of smallpox, a devastating disease that once killed millions of people worldwide. They have also significantly reduced the incidence of other serious diseases, such as polio, measles, mumps, rubella, and tetanus. By continuing to vaccinate against these and other diseases, we can protect future generations from the devastating consequences of vaccine-preventable diseases. Vaccination programs rely on ongoing surveillance, research, and public education to ensure the safety and effectiveness of vaccines. By working together, we can continue to harness the power of vaccines to protect individuals and communities from infectious diseases.